some of the diagrammatic expressions of the details that we can discuss it nicely through this presentation. So I'm sharing my slide and I'm giving it the full screen mode. I hope all of you can see my slide.
Yes, sir.
Okay, fine, thank you. In your syllabus, no doubt they have given it is a Circoviridae, and for quite a long period, even if you pick up your textbook, little bit few years back textbook, then you'll find only the Circoviridae family and important disease, Porcine Circovirus and second infectious Anemia virus infection. They have discussed under the Circoviridae, but recently it has been seen that the second Anemia virus is no more closely related to the circoviruses. Rather, they have been grouped into another family called as the Anelloviridae. So, in today's presentations, we'll be discussing Circoviridae and Anelloviridae. No doubt, both these group of viruses extremely related and almost similar characteristics, but genetic difference is there. So before going to this family, let me tell you, these are the viruses which are till now considered to be the smallest vertebrate viruses. Circovirus, Circo and the Anellovirus, these are the example of smallest vertebrate virus. They are the DNA virus. Beforehand, in our previous classes also I mentioned about the picoRNA. Pico means small. RNA virus, small RNA virus that include this foot and mouth disease that we have discussed it. Then we talk about the DNA under the DNA group, the parvoviruses, parvus, the parvoviridae family derived from the name parvus. Parvus means small. So because of their small size of around 25 to 30 nanometer, they were considered to be the smallest viruses. But when the Circoviridae members were identified and they were known to cause certain pathological consequences. In fact, it is becoming a very emerging disease and causing a major economic losses in poultry as well as in the pig in terms of producing a sort of immunosuppression and total production loss in the animal. And gradually throughout the globe, people are recognizing it as a major issues, and the efforts are made to eradicate or remove the infections from the farm premises. This is some sort of very silent type. Silently they hit the farm, and subclinically the animals may develop these infections, and they may cause immunosuppressions, which many times become unnoticed to the people because of no very much typical manifestations. So, but it can weaken the immune system, and that may be the reasons for vaccination failure. That may be the reasons for many secondary viral infections that flares up in the pig as well as in the poultry. So as such, this particular virus is now recognized to be a major organisms which we should concern, and in terms of animal health, this is really important for us. So for quite a long time, people never ignored this virus, although it was detected long time back in 1970s itself. But at that time, it was thought that they are non-pathogenic, normal microbiota of the animal. But later on found that few members are potential pathogens and they are associated with the disease. So circoviruses are now we considered as the smallest vertebrate virus, and their size is almost 17 to say 20 nanometers. Some members a little bit bigger size, like 23 to 26 nanometer also we see, but the smallest one identified to be 17 nanometer. So it's an extremely small particle, virus particle, and now we considered them as the smallest vertebrate virus. Okay, and a second important points with the circoviruses, every one of you will identify that characteristic is that they are the example of double-stranded RNA, the single-stranded DNA virus. So in nature, all the DNA they are double-stranded, but this Circo and the Parvo viruses, they are the example of DNA virus having single-stranded genome. Over and above this Circoviridae, they are called as Sarco because of a circular conformation. This circular conformation is not the virus conformation, it is the circular genome. The Circovirus single-stranded DNA, both the ends are covalently closed. As a result, their genome is closed and circular, just like a bacterial plasmid. And in fact, they replicate by the rolling circle method as the plasmid replication takes place. So it's quite interesting. Again, you'll find this virus, they are having extremely small genetic material. This is really a fascinating points in the field of virology, like how they can be called as an organism with such a small genome. The genome size is 1700 nucleotides to around 2200 nucleotides. So it's extremely small genome size, and they adopt certain strategy of transcriptions, which is, we know that with a single open reading frame, they can code the proteins by overlapping fashions. That in some slides we'll try to explain it. I hope you will understand that point. And using the small genetic information, they can code for some proteins, but protein numbers is also very, very less. Hardly 3 to 4 proteins they coded, and they are happy with that, and they can survive in the nature. So this is some sort of an evolution of the virus and their existence in the nature. So these are some of the size of the circoviruses, and the circular genome through electron microscopy has been focused. So they are called as the circoviruses because of the circular conformation of the genome. Please note it down. It is not because of a circular type of virus. Of course, their icosahedral symmetry, the triangulation number is 1, icosahedral symmetry. They have around 60 copies of the capsids in total. So the viruses of our importance, of course, there are many members identified under this particular family. So Circo and the Cyclovirus, these are the two genera under this Circoviridae family. And where we'll be discussing about the Circovirus, that include the Porcine Circoviruses. And Porcine Circovirus is a disease that we are going to discuss under this particular topic or in this particular family. But you must understand the general characteristics of this group of viruses. So with such a small genome, how they can survive in the nature. And probably this is opening a new field in the in molecular biology, where people are thinking of utilizing this particular virus backbone as an vector. And a lot of processes are going on to prepare some vector to vaccine using the Circovirus as a backbone. Okay, fine. So coming to the general characteristics of this particular family, then you'll find here having the icosahedral symmetry, and the triangulation number is 1, T is 1, T number is 1. And this particular virus, they replicate in the nucleus. They essentially need the host cell polymerase enzyme, the DNA-dependent DNA polymerase to initiate its replication. And as you say that the genetic material, it is extremely small. It's hardly few hundred, 1700 to 2200 nucleotides. That makes the entire structures of the genomic component of the circoviruses. So it's very hard to believe how an organism with such a small genetic information can survive. So definitely, it's we should call it not an organism. Rather, we should call it a virus particle. So they adopt certain very special transcription strategy or agenda they have to survive by overlapping transcription mode. where the overlapping sequences, they can code it, and having only two open reading frame through which they can code the protein. So they replicate the nucleus, and this is another most worried point for all of us. These virus particles are extremely stable in the environment. I should use the word extremely stable as because you can see 56 degrees centigrade up to 1 hour it can withstand. So it's a very stable virus in the environment, and the disease transmissions always occur by lateral mode of transmissions. That means the infected animal excrete the virus, and in contact animal pick up and they develop the disease. Okay, so this is a highly stable virus. Then if you talk about this, the other propagation of the virus, it can be propagated in implanted chicken egg for diagnosis purpose. However, in conventional cells, it is sometimes difficult. They very much prefer the bone marrow cell. So there is a technique called as bone marrow cell propagations, where we can isolate this virus very easily. They had tropisms towards these bone marrow cells. So very quickly, we'll just see it here. Then the virus through this receptor-mediated endocytosis will enter, and then the entire replications will take place inside the nucleus. They totally depend on the host enzyme system for initiation of replication. Then what you can note it down, the DNA replication is by rolling circle method. That is common for the bacterial plasmids and bacterial chromosomal replication. Okay, and the protein synthesis that takes place in the cytoplasms, and thereby the assembly also takes place in the cytoplasm. The new mature virus particles, they escape the cell. So in this process, in the circovirus infection, they produces very prominent intracytoplasmic inclusions, which is diagnostically important, right? So this understand get aggregated, or maybe some degenerative cellular pods get aggregated, and that will give rise to the in the inclusion body formation. Just in a very simple picture with 1.7 kb size of the circoviruses, they had only one, two open reading frame, and they are having only one promoter regions. In both the way, they can use it for coding for capsids and the replicase enzyme. So with very little amount of protein, this particular virus can exist in nature. These are some of the general characteristics. So coming to the specific infectious organisms, the first is that the porcine circovirus infection. For quite a long period, people were it was remained unnoticed for the pig husbandry. People who are in the pig husbandry practices about the importance of this infection. So the first member, porcine circovirus 1, which is considered to be a cell culture contaminant virus through certain pig vaccines, which is created in the PK15 cell. So the vaccine virus propagating in the PK15 cells, somehow in 1974, it's found that there is an existence of a cell culture contaminant and identified as a circovirus. And that particular one, porcine circovirus 1, where seems to be an non-pathogenic, and in fact, they do not causes any disease condition in the animal. Another variant, genotypes of the porcine circovirus were identified, is called as porcine circovirus 2. Now, it has been seen that this porcine circovirus 2 is associated with pathological consequences, and mainly it causes acute immunosuppression in the animal and responsible for a wasting type of condition. Gradually, the body weight losses will take place, and the animal will never attain to its market weight if it's having this circovirus 2 infection, PCV2 infection. So, porcine circovirus 2 is the main organism that is causing this disease conditions in pig. However, gradually in the timeline, if you see in 2015, another porcine circovirus 3 were identified, and are associated with the same kind of conditions as PCV2. And very recently, porcine circovirus 4 has also been reported from certain country like China as well as in the USA. They reported some novel circovirus, porcine circovirus 4. So likewise, if I've been asked that the porcine circovirus, the causative organism, then you have to mention it is the porcine circovirus 2. Okay, 3 and 4 yet to be established as an major pathogens that causing this particular infection. However, these are the different members identified under the porcine circovirus family. Okay, so PCV2 is the major causative organisms, and a lot of genetic variants were identified, like 2A, 2B, 2C. So newer and newer informations are coming to the scientific community about the disease of porcine circovirus. Classically, this the symptoms what recorded caused by the PCV2 infection is sort of a post-weaning multi-systemic wasting syndrome. Shortly, we call PMWS. Please note it down. Post-weaning, usually seen after the weaning only the clinical manifestation, and that basically causes a multi-systemic wasting syndrome. Means gradually the weight loss will take place, and there will be acute immunosuppression in the animal, and certain systems like kidneys are affected, and even the dermatitis conditions that because of the clotting factor defects and secondary bacterial infection they invaded. This is what we call porcine dermatitis and nephropathy syndrome, PDNS. These are different clinical manifestations of the same disease, commonly we call as a porcine circovirus infection. And the causative organisms, right now we say porcine circovirus 2, 3 variants, 2A, 2B, 2C. However, some recent report says that this type of syndromes also associated with porcine circovirus 3 and 4. These are different genetic variants identified till now. So these organisms till now it is found to be restricted to the pig population only. They are the natural host, and the worry point is that lateral transmissions occur, and very easily an animal may pick up the infection, and the infected animal excrete the virus, and thereby laterally it may transmit. So it is 2020, another report from the porcine circovirus 4 from Hunan province in China. So likewise, okay, this particular picture is always I favor to make you explain what actually is circovirus infection, porcine circovirus infection. Just look at these two pigs. Both are from the same litter. Both were reared on the same nutritional regime. The smaller one is an PCV2 positive, and the other one is negative. So this is the situation what it occurred. The animal appetite, everything is seems to be absolutely normal, but there will be no growth or growth, and the animals will show some sort of wasting kind of syndrome. There are some other pictures like this is a particular flock. This animal is having PCV2 infection in comparison to the other. So this is the typical things that happen in the porcine circovirus 2 infection. This is not an infection where you can see a large scale mortality occur within a very rapid short period. However, this is considered to be an epidemic right now, that it is spreading to many flocks throughout the globe, and gradually it is weakening the immune systems, and the animals are vulnerable for many other type of infections. So although the mortality is not directly because of this one, but the infection itself causes tremendous economic losses in the farm, and because of poor growth, and the animals are susceptible to many other infections. So this particular slides what I want to show about the very prominent intracytoplasmic, intracytoplasmic inclusions, which is a characteristics of porcine circovirus 2 infections. These are some of the inclusions showing it here, very prominent inclusions, which are diagnostically important for us. Okay. Now, if we try to discuss about the pathology, what actually this particular virus do, then you will find they mainly target the lymphoid cells, particularly the bone marrow cells are the target. This is again somewhat similar to that of the parvoviruses. In the parvovirus family, what we discuss is that parvoviruses, they prefer to grow in the actively dividing cells. The reason so is that they don't have the polymerase enzyme, and they totally depend on the host cell polymerase enzyme for replication, and the host cell DNA polymerase enzyme abundantly present in the actively dividing cells, not in the resting cells. So here also the bone marrow cells, where these are very actively dividing, are the target for the circoviruses. Secondly, they target the lymphoid cells so that they can establish their existence inside the animal. Because this virus, the capsid proteins are so strong that the immune system can readily eliminate the infection. So they target the immune cells and immunosuppression occurs, and the virus can survive for a longer period inside the animal body. So the primary target is the lymphoid cells, and there is acute depletions of this lymphoid cells. Both lymphoid and myeloid cells are affected. But basically, more striking feature is because of infection of this kind of cells, like they can target the interfollicular dendritic cells. You understand what will happen if the interfollicular dendritic cells will be targeted, and if they will undergo degenerations in the form what we call as the apoptosis. This particular virus can induce apoptosis in the cell. They specifically target the B lymphocytes. So obviously there will be humoral immune response will be suppressed. NK cells, which are one of the sentinel cell inside the animal body, which can non-specifically kill any kind of abnormal behavior happening inside our body because of the virus infections. The natural killer cells plays a very, very important role in giving the early protections against the viral infection, because non-specifically they are not in the adaptive immunity, they are innate immunity brands, the NK cell, natural killer cells. But the circoviruses target these NK cells, and they can induce apoptosis or apoptotic cell death, enhance the NK cells elimination from the body. Then gamma delta T cells. This is another target of the circoviruses, porcine circovirus. You know, the alpha beta, the CD4 and CD8, the helper and the TC cells, which are the part of the adaptive immune system. However, the gamma delta T populations, which comprises almost 15 to 20% of the total T lymphocytes, this gamma delta, they also behave just like the natural killer cells in a non-specific way. They can causes the virus elimination from the body. This gamma delta T cells are also a target of the porcine circovirus. Now, what overall idea you can have is that this virus, they specifically target the lymphoid cell populations. As a result, there will be immunosuppression. Okay, so that is clinically reflected by some kind of symptoms like wasting syndrome, pallor of the skin, then anemia will develop, respiratory distress and diarrhea, jaundice. These are different consequences that you can see, but gradually it appears in the animal. Probably this is the reasons why for quite a long period people are unable to understand that this is a viral infection going on in the animal. So later on it was established, and this new genotypes like VCB2 are producing some appreciable clinical manifestation, because of which nowadays it becomes easier for diagnosis purpose. So certain study says that usually affect in 2 to 4 months of age, just after this weaning, and the morbidity is of 4 to 30%, and mortality may goes up to 20% as recorded. And this mortality is basically because of secondary bacterial infections in the animals, and the death occur. Okay. So interstitial pneumonia, enlargement of the subcutaneous lymph node, these are some of the clinical manifestations that we can see. The porcine dermatitis and nephropathy syndrome, this is another clinical manifestations seen in certain countries. This is because of the defects in the clotting factors, and then secondary bacterial infections, maybe the Clostridium as well as the Staphylococcus organisms, they may causes this kind of lesions. And the immune complex circulating in the body can also cause the nephritis situations. Okay, it's called as PDNS. That's about the Porcine Circovirus infections. But how it is important for us is that they make the animal susceptible to many other infection because of this immunosuppression. So for diagnosis, no doubt the poor growth and the conditions gives us some information, but these are not concrete enough to suspect about the circoviruses. So always it's on the detections of the antigens in the clinical sample, as well as the serum antibody screening can give us information about the prevalence of the disease in a particular flock. Okay, so for that we need to collect these clinical samples that obviously include the blood, the PBMC cells, peripheral blood monocytes as a source of the virus. We can collect the lung tissues after postmortem, tonsil, spleen, lymph node. These are the things, including the bone marrow. Bone marrow can also be collected for specific detection. One of the very easiest technique for rapid detections, which are followed in many laboratories, is that FAT test. Straight away, they go for this FAT test. This is a slide I'm showing. This is an impression smear from the bone marrow soup, and that the fluorescent antibody test using the FITC conjugate, where stained and the infected lymphocytes are showing this fluorescent activity. So very rapidly, using an immunostaining, we can detect the virus. However, we can go for the traditional way of isolation and identifications, and by the secondary test, like immunoperoxidase test, they are not very much cytopathic in the cell culture. So we have to depend on the secondary methods, either the FAT or immunoperoxidase test and the monolayer we have to perform for detection of the virus infection. However, as you know, beside this all antigen antibody detection test, the PCR plays a very important and rapid approach for quick identification of the organisms. And not only identification of the organisms, even we can find out the types, the particular types of the virus, whether it is a PCB2A, 2B, 2C specific PCR can be performed and it can be identified. Okay. So have you ever come across this disease anywhere, anytime? Hello? Please respond. Hello? Please respond.
Yes, sir.
Yeah, have you ever come across these conditions in the pig? Not very specific, not specifically. Now onwards, whenever you visit, try to see these aspects also. I'm not sure about in the Nagaland. Yes, from Nagaland also. One of our PG students from pathology department, he screened and isolated this virus from state of Nagaland, and lot many isolates from Mizoram. So this is very well prevalent in this area, and almost everywhere they are now detecting this circoviruses, and they are some sort of an hidden type of an infection persists in the populations, and that is now it is considered to be a sort of epidemic. So recently, the Ingelvac has come up with an vaccines, and they launched this particular vaccine in the Northeast India, and this vaccine is available in the market, and it is an inactivated, and the approach is an virus-like particle. They actually synthesize the capsid protein of this particular virus, and auto-aggregation of the capsid protein gives rise to a VLP, virus-like particle. It is an inactivated, totally inactivated, without having any genetic information. they mix up with certain suitable adjuvant, and they claim that this can give very good protections against this circoviruses, a single shot vaccines. Even they suggest if the whole flock of some number of animals are positive, then also we can administer these vaccines, and that vaccine is available. It is a VLP-based vaccines in Northeast India, as I know. So these are some more information about this Circoflax in Zelvac companies, and this include this VLP of Circovirus 2. And please go through this particular dosings and immunity from the literature provided by the company. And we should go for vaccinating the animals, as because it is appearing to be a silently killer disease in the pig. And that's about the porcine circoviruses. Now I'll be concentrating to the second family, the Anelloviridae. Still students are admitting. I don't know why you're so late. Okay, fine. The next family is the Anelloviridae. Of course, it is not mentioned in your syllabi, but this is the things you must understand. The second infectious anemia. The disease has been named there. Earlier, it was discussed under the Circoviridae family, but recently it has been totally separated from the circoviruses as having certain genetic differences. A separate group of virus, they name it as the Anelloviridae family. Here also the same meaning. The Anello means a ring structures, round, circular genome, the same as the circo. So this particular family, different members were identified. This is on the electron microscopic picture. Very small, say 19 to 26 nanometer is the size. Icosahedral, all characteristics are similar. However, this Anelloviridae, the size of this genome is a little bit bigger than that of the circoviruses. Circo we have seen 1.7 to 2.2, but here 2 to 3.9 kb. So they quote a little bit more genetic information. However, this information is extremely small information. Mind it, extremely small information. It is difficult to believe how an creatures can have such a small genetic information and can give rise to new virus particle. So this is a very fascinating field of virology. So this again gives us many clues and newer information in the biology, how the transcriptions, different types of transcriptions may occur in this the process in this group of viruses. So we'll come to that point again in the next slide. So similar to that with circoviruses, they are again very stable in the environment, and the transmissions may occur both laterally as well as vertically. That's a worry point for us. So this viruses, Anelloviridae, they can be propagated in the lymphoid cell line. So the other cells, it's difficult to grow them. So the all the replications and remnants can be provided by the cells of lymphoid origin. So as such, certain some blastoma cell lines are available which are used for identification. Okay, this is not much important, just to show you the phylogenetically how they are different from the circoviruses. That's why they have been grouped under the gyrovirus genus, and that is the second infectious anemia virus. So this is the only disease we are going to discuss. The chicken infectious anemia, it is an already known disease, and it is considered to be have severe economic losses in the poultry industry, I should say, and it's rampant in our country. Almost in all states, they recorded this infection. Even in the state of Mizoram also, we could detect this particular virus, second infectious anemia virus. So from name, what we can idea that the profound anemia occur, and this is highly infectious, means transmissions is rapid from one bird to the other. And basically, you'll find they again target the hematopoietic precursor cells. As a result, the marked depletions of the cells takes place. So it was first identified in 1979 in Japan, but thereafter spread it to many countries, and our country is detected almost from all states. And this is an important viral infection in the poultry, targeting the causing the immunosuppression. So the causative organism is avian gyrovirus 1. Earlier it is called as second infectious anemia virus. Both the names are valid, I should say, because this is a very new nomenclature. And the genus is the gyrovirus, and the family is the Anelloviridae. So don't get it confused. In some textbook, you'll find it's a Circoviridae family and chicken infectious anemia virus. The causative organism they will write. No matters. So this is the latest classification. If you have any doubt, what you will do? Simply will go to the ICTV website. I taught you how to go through that websites and how to make it sure that the latest classifications in the taxonomy tab, you can go through it and you can identify it. Now coming to the host, you'll find a sequence of all ages they're susceptible to virus infections. So as this animal ages increases, as you know, this group of viruses, they are very good immunogens. So the immune system, if it is strong enough, they can elicit the antibodies and they can neutralize the virus infection. So susceptibility decreases as the age progresses. Okay. This is the most worried point, the vertical transmission. So this infection, both horizontally and vertical transmission occur. So horizontal transmissions, we can prevent it by adopting different measures of containment and strict biosecurity and by disinfecting procedure. That is at our hand. But vertical transmission is again matters, because most of the times our farmers, they purchase the chicks from certain vendors, some producers, and if the parental flock is having the infections, then vertical transmissions may occur. So I have to take care about it. So this is a picture showing these 3 proteins produced by chicken anemia virus, VP1, 2, and 3. And these are all overlapping. You can see 3 and 2 are within the same genomic loci. And under the influence of single promoter, these transcriptions occur. So this is the genetic material, this particular part, that coded for VP2 as well as VP3. Even the VP1, some part is the overlapping. So this kind of strategy adopted by this viruses having very small genome size, so that more they can code from the less amount of genetic material. That is the logic. Okay. Here, this VP3, this is considered to be this VP3 is a capsid protein. This is responsible for causing the main pathology, and this VP3 protein is also called as apotin. Apotin, please note it down. Please note it down. The VP3 protein of second infectious virus, anemia virus, is called as apotin, and this can induces apoptosis in thymic lymphocyte. This is the main pathology that causes the depletions of the cells. You know, the apoptosis is a normal process. Every cells, when there is virus infection, there is a tendency to cause apoptosis. But producing certain protein, VP3 protein, which are mainly act against the tumor suppressor proteins, and they can cause rapid apoptosis process, and they eliminate the bone marrow cells. So this virus, through oral route, they enter, and they had hematopoietic precursor cells and thymic precursor cells, and where they causes apoptosis, induced apoptosis that leads to severe immunosuppression. Let us try to see these particular pictures. These are the target. You know, like inside the bone marrow, we had a pluripotent stem cells from which the lymphoid and the myeloid progenitor cells they get differentiated, and that the lymphoid progenitor will give rise to all T B lymphocytes, including the NK cells, gamma delta T cells. These are produced, and the myeloid, all other blood cells are produced, granulocyte, thrombocytes, erythrocytes. So the target you can notice here. is the hemocytoblast. Before differentiating to this Porcine, they will infect second anemia virus and will undergo apoptosis. These cells will undergo apoptosis. As a result, marked depletions of hemocytoblasts will occur. So the resultant thing is that all these cells will be depleted. As a result, there will be acute immunosuppression. Erythrocytes depletions will lead to severe anemic condition. Thrombocytes will give rise to defect in the clotting factor. The same way in the lymphoid precursors, if you say that the T lymphocytes precursors are targeted, then there will be acute immunosuppression. As you know, the T helper cell is the prime cells that can support the B cell for differentiation. So likewise, this particular virus, they mainly target the precursors, hematopoietic cells. As a result, there is marked immunosuppression and severe anemia produced in the animal. And these are the clinical manifestations of what we could see. So up to 60% mortality and atrophy of the lymphoid organ, etc., etc. And the blood pictures will immediately tell you. There will be stunted growth, poor growth after giving good feed is an indication in the poultry farm that the flock might have this infection. However, there is another, this is a gangrenous dermatitis condition in the poultry. Good to see here. Basically, this is multifactorial. One is that the clotting factor defects leads to accumulation of blood clot anywhere on the body surface, and particularly the wing slide injury will lead to development of hematoma, and this type of clots are developed. And later on, it is invaded by the common saprophytic bacteria or even certain pathogenics like the skin normally presents like Clostridium septicum, then even Perthingens A, the environmental which are abundantly present, maybe the Staphylococcus aureus itself. So why they will get the invasions? One is the breeding, that the blood clots will be there, and secondly, the bird is immunosuppressed. So as a result, they can induce some sort of gangrenous dermatitis condition. So this is very frequently seen, and we can get some idea. The primary cause is the second infectious anemia virus infection, not the Clostridia or Staphylococcus. These are some of the symptoms that we can see. So thereby we can get idea about the disease. So for diagnosis approach, again, the clinical signs and symptoms, of course, will give us some information about this poor growth conditions, and then severe anemic conditions, pale mucous membranes and combs and wattles that can be reflected. Then poor growth is one of the features, and gangrenous dermatitis if some animals, birds are showing. But this is not conclusive, so have to confirm by identification of the organism, by isolation and identification. So obviously we have to collect this different samples for detection. And as I say that we need certain specific lymphoblastoid cell line where we can grow this virus. And usually they are not cytopathic, and we need to use the secondary methods like FAT or immunoperoxidase test for its detection. But there is a system of embryonic chicken egg for isolation of the virus. And primary bone marrow culture, as I said, that they are the main target. These are some primary cells, and they can be propagated in the laboratory flux, and then we can give the virus infections for isolation purpose. Then again, here also the molecular test, PCR-based test gives us very rapid and accurate information about the disease. Very easily with minimal facility we can perform it. Okay. Coming to the control of this infection, so some sort of vaccines, chicken anemia virus vaccines, modified live virus vaccines marketed by the Merck. However, the availability is not so in our market, particularly in the Northeast India, what I should say. However, it can be procured. It is licensed in our country, and this particular vaccines can give good protections against the infection. And usually this vaccine is given by the wing wave inoculation method. I have shown you the video how this wing wave inoculations of smallpox vaccine has been given in our earlier discussion. And they gives us protection. However, we must remember that the disease may come through vertical transmission. So as a result, it's always needed to make sure that the parental flock doesn't have these infections. Even in certain state animal husbandry department, they maintain their breeding flock, and chicks or hats distributed to other area. So one have to make sure by either serosurveillance or by detecting certain suspected bird with all these techniques, whether the flock is disease-free or not. That's quite important, and we must promote the detection of this chicken anemia virus in your area. So these are some informations that I shared with you. And if you have any queries or questions, or if you can want to supplement more, I'll be very happy. Yes, do you have any questions? Again, I'm repeating, yesterday class also I told you, like for the final year students, we are conducting the training program for JRF examinations. And mind it, in JRF examination, they ask basically this kind of questions only: the appropriate routines of chicken anemia virus. They will ask about the circular genome virus. These are the things they generally ask in the examinations, in competitive examinations. So at this point onward, if you prepare your own questionnaires and essays and makes your own notes properly, if you keep it, definitely it will help you at the time when you prepare for the examination. So please keep in your mind, and yeah. Do you have any questions or other things we can share and we can discuss? 38 students have joined. Very poor numbers. I don't know what happened to our student. Very few